Hello, everybody. My name is Danielle Rock, and today I'll be telling you about my undergraduate research at Trent University on inbreeding and inbreeding depression in the invasive hybrid cattail Typha x glauca and its progenitors. To begin, I will go over some background information just to help set the stage for my research. So what is a cattail? Cattails are those green plants with the brown, furry, hot dog-looking fruits on top that you can see along roadsides and in wetlands in the summer months. They are an obligate wetland species that is essential for the proper cycling of nitrogen and carbon within their habitats and also provide shelter for many microorganisms, insects, and birds. They can typically be found in ditches along the roadside and in wetlands, preferring water that is less than 3 meters deep. Here in Ontario and around the Laurentian Great Lakes region, there are 3 prominent taxa: the native Typha latifolia, the introduced European Typha angustifolia, and the hybrid form between the two, Typha x glauca. Here we can see an example of each of the 3 taxa. In the top left is an image of the native Typha latifolia, and in the top right is an example of the introduced European Typha angustifolia. In the middle at the bottom is an example of the invasive hybrid Typha x glauca. The native Typha latifolia and introduced Typha angustifolia make up the parental species of the hybrid. When the pollen from latifolia fertilizes angustifolia, they produce their hybrid offspring, Typha x glauca. This usually occurs where the 2 parental species can be found coexisting, an occurrence typical of the Laurentian Great Lakes region. These hybrid cattails are currently displaying various invasive characteristics around the Great Lakes region. They show heterosis, which is the increased fitness of hybrids relative to their parental species, and are expanding in their habitats at the expense of these parental species. This is having detrimental effects on wetland habitats, mainly in the form of a loss of diversity, as multiple species are being pushed out and replaced by stands of the hybrid cattail. Cattails can reproduce both asexually and sexually. Asexual reproduction occurs through clonal reproduction, which essentially occurs when the parental plant grows a vegetative module, called a ramette, producing a genetically identical clone of itself attached through underground connections. My study will only be focusing on the forms of sexual reproduction, though it is important to note that clonal reproduction does still occur naturally. The first form of sexual reproduction, seen in the graphic in the middle, is outcrossing, which occurs when the pollen from one individual fertilizes the female inflorescence of a separate individual. The second form of sexual reproduction, seen on the right, is self-fertilization, which occurs when the pollen from an individual fertilizes either its own female inflorescence or the female inflorescence of a clone mate. Since self-fertilization is a form of inbreeding, it typically leads to increased homozygosity, as both of the genotypes coming together are from the same individual or clone mate. This can lead to inbreeding depression, which is the reduction of an individual's fitness as a result of that increased homozygosity. This homozygosity is typically higher in the parental species compared to the hybrid, as the hybrid is formed by the mating of 2 distinct, separate species with distant genomes, which is going to make them more heterozygous in nature. To help explain this, let's go through a thought experiment. On the top, I have an example of self-fertilization in the parental latifolia. As you can see, the paternal and maternal alleles coming together are genetically identical, as they are coming from the same individual. This individual is homozygous at the A and C loci and heterozygous for B. Following self-fertilization, the resulting offspring are going to most likely display an identical genotype to the parental species. However, due to the heterozygosity at the B locus, there are possible combinations that can create some genetic diversity. In contrast, on the bottom, I have an example of a hybrid undergoing self-fertilization. Once again, the paternal and maternal alleles are genetically identical, as again, they are coming from the same individual. However, this time, the individual is heterozygous at each of the 3 loci, which is a common occurrence in those hybrid species. Following self-fertilization, again, the offspring are going to most likely display that identical genotype to the parentals. However, due to the heterozygosity within each of the 3 loci, there are more possible genotypes for the self offspring of the hybrid compared to the possible genotypes for the self offspring of a parental species. As there are multiple reproductive strategies used by cattails, there are multiple routes that can lead to inbreeding. These include the clonal reproduction that produces genetically identical clones, outcrossing between plants that are related, and self-fertilization. Earlier studies have quantified inbreeding in cattails, finding that the inbreeding coefficients are high for each taxa, as seen in the table on the right. This leads to the assumption that inbreeding is occurring in natural cattail stands. Despite this, there has been no exploration of the effects of inbreeding on these Typha species. All of this information coming together leads to the question of what affects the reproductive success of hybrid cattails in the Laurentian Great Lakes region. Using the information provided, I hypothesized that inbreeding affects this success and predicted that due to that increased heterozygosity in hybrids, inbreeding depression would be less pronounced in their self offspring than those of the parental species, as again, increased homozygosity typically leads to a greater magnitude of inbreeding depression. So, how did I go about actually testing this hypothesis? I conducted field sampling in the summer of 2020 using 18 different sites in and around Peterborough and Selwyn County, as denoted by the red markers on the map. I ensured each site had a minimum of 2 maternal plants and a maximum of 40, half of which I would make outcross and half inbred. At the end of my project, my sample size was 231 maternal plants, the breakdown of which can be seen in the table on the right. I ended up with 76 total latifolia, 67 angustifolia, and 88 hybrids. For outcross plants, I collected pollen from non-maternal plants at the sites. I did this by shaking the male inflorescence into a glassine bag until there was a substantial amount of pollen collected. I then painted the pollen onto the female inflorescence using a 1-inch angled paintbrush, ensuring total saturation of the inflorescence, meaning I essentially painted in up and down motions all around the inflorescence until the little hot dog fruit turned from brown to yellow. I then rebagged the plants to help prevent cross-contamination from surrounding pollen that was dispersed by the wind. I followed a similar protocol for inbred plants, except instead of using a paintbrush to apply the pollen, I simply broke off the male inflorescence and rubbed that directly onto the female inflorescence until again it turned from brown to yellow. After I collected all the pollinated fruits in the fall, I extracted the seeds from them. I weighed out 0.5 grams of the seeds and separated them from their achenes, which is essentially just that fluffy white bit that allows the seeds to disperse through the wind. I then put the extracted seeds in petri dishes, submerged them in DI water, and counted them. Next, I left the seeds to germinate in the greenhouse for 10 days, after which I counted the number of germinated seeds, which was essentially just any seed that had any sign of growth, so whether it be just a little itty-bitty white root tip poking out or a full green shoot emerging as seen in the bottom right image. Using all of this information, I calculated the germination rate by dividing the number of germinated seeds by the total seeds extracted from 0.5 grams. I used ANOVA to compare the mean values for seed set and germination, essentially comparing each treatment group. For example, I would take inbred latifolia and compare it to outcross latifolia, and then inbred latifolia compared to outcross angustifolia, etc., to determine which were significantly different from each other. I use these early life measures as proxies for fitness to infer the presence and magnitude of inbreeding depression. Now let's explore what I found. There were no significant differences between the inbred and outcross seed set in any of the 3 taxa. Looking at the graph on the slide, we have angustifolia on the left, the hybrid in the middle, and latifolia on the right. As you can see, the pink inbred and the blue outcross values within each taxonomic group are not significantly different from each other, meaning that there was no indication of any fitness differences between the mating types, and thus no indication of inbreeding depression for seed set. However, denoted by the letters at the top, you can see that there were taxonomic differences. This means that the inbred and outcross values within each taxonomic group were significantly different from those values in the other 2 taxonomic groups. There also were no significant differences between the inbred and outcross germination rates for any of the taxa. Looking at the graph on the slide again, we have angustifolia on the left, hybrid in the middle, and latifolia on the right. And again, as you can see, the pink inbred and blue outcross values for each taxonomic group were again not significantly different, therefore finding no indication of inbreeding depression for germination rate. Once again, denoted by the letters at the top, there were taxonomic differences, which again means that the inbred and outcross values within each taxonomic group were significantly different from those in the other 2 taxonomic groups. Overall, there was no evidence of inbreeding depression at the early life stages, as the inbred individuals did not have significantly different fitness from the outcrossed. However, there were taxonomic differences which may indicate differences in fertility between the taxa. So now, what does this all mean and what are the next steps? I failed to find any support for my hypothesis that higher heterozygosity in F1 hybrids results in a lower magnitude of inbreeding depression, as there were no significant differences between the inbred and outcross fitness, and thus no evidence of inbreeding depression in early life stage development. This also means that the prediction that the increased heterozygosity in hybrid offspring aids in suppressing inbreeding depression is not supported, at least in the early life stages that I explored. Unfortunately, this data does not help in our understanding of why the hybrid is dominating wetlands around the Laurentian Great Lakes region. So now, let's explore some possible explanations for my results. One likely explanation for my results is genetic purging. Genetic purging is the removal of deleterious alleles in a population via natural selection, which acts to prevent inbreeding depression. This is a common phenomenon in populations which reproduce predominantly through self-fertilization, and to protect that population from extinction, natural selection works to remove deleterious alleles in early life stages. So essentially, those seeds which had deleterious alleles expressed in their genome in early life stages would simply not germinate. This likely applies to my system, as it has been shown that there is a history of inbreeding in both parental species, as evident through the inbreeding coefficients I presented earlier in my presentation, and the multiple routes that can lead to inbreeding. However, it is important to note that deleterious alleles that are only expressed in later life stages of cattails would not be removed by purging in my study, as these genes would simply not be turned on in early life stages, and thus may be expressed later and result in later life inbreeding depression. My findings may also be a result of hybrid sterility and breakdown. Sterility is when the reproductive capacity of hybrids is suppressed. So basically, the hybrids are less likely to produce their advanced generation offspring due to genetic inviability. This could be an explanation for why my hybrids had significantly lower seed set than either parental species. There also may be hybrid breakdown within cattails, which is essentially when the parental species have an increased fitness in comparison to the advanced generation hybrids, which was evident in my data through the taxonomic differences in germination rate, where those F2 hybrids had the lowest overall germination. This concept is common in hybrids, as the wacky genetics from the first generation hybrid offspring get copied over into the advanced generation hybrid offspring and result in a lower relative fitness. So using what we know based on my data, where should we go from here? A really great place to start would be to explore the presence and magnitude of inbreeding depression in those later life stages. So grow the plants to a mature stage and use the plant height and biomass as predictors of fitness and compare those in the inbred and outcrossed individuals. This is important to do because evidence points to the majority of self-fertilizing plants typically displaying inbreeding depression in later life stages. It also is important to explore whether these findings are regionally dependent or whether they are consistent across all of Ontario. This could help shed some light on the differences in hybrid dominance across the regions, either by eliminating the factor of inbreeding depression as contributing to dominance, or providing an explanation as to why it is dominating wetlands around the Laurentian Great Lakes region specifically. My research is still very novel within the field, and thus is a preliminary exploration of the effects of inbreeding. There still is a lot of work that needs to be done before any concrete, conclusive statements can be made about inbreeding depression in cattails. So to conclude, there was no indication of inbreeding depression at the early life stages in any of the 3 cattail taxa. This was determined using seed set and germination rate as proxies for fitness and comparing them in inbred and outcrossed individuals. This may be a result of genetic purging, removing deleterious alleles before inbreeding depression could be detected in early life stages, and thus, future research should explore later life stage effects and regional differences to help get a more holistic picture of inbreeding depression in Ontario cattails. To finish up, I would like to thank a few amazing individuals without which this project would not have been possible. First, I would like to thank my supervisor, Dr. Joanna Freeland, as well as my co-supervisor, Dr. Marcel Dorken, for their overwhelming support both academically and financially through this project. I also would like to thank Vikram Bhargav for his help conducting fieldwork and running my stats in R, as well as Christina Steinak for her help with my DNA analysis. Without this four, this research would not have been possible. And with that, I would like to thank all of you for listening and look forward to discussing any questions you may have afterwards. Thank you.